Hi friends, again welcome to UNS classes. We are back with another previous year NEET question. Today the question is a multiple choice question and we have to find out which given sentence is wrong. So let's go to the question. Here the question is, which of the following is an incorrect statement? Option A is nuclear pore act as passage for proteins and RNA molecules in both directions between nucleus and cytoplasm. The second option is mature sieve tube elements possess a conspicuous nucleus and usual cytoplasmic organelles. Third option is microbodies are present in both plants and animal cells, and the final option is the perinuclear space forms a barrier between the materials present inside the nucleus and that of the cytoplasm. Right, all these are related to the topic cell. So as usual, let's go little by little detail about each option and come to the answer. So the first option is about the nuclear pore and the last option is about nucleus only. Both are related directly to the nucleus. So let's see a little bit about nucleus. So what's a nucleus? A nucleus is nothing but a nucleus is called, what a nucleus is called in a cell? It's called the director of the cell. Director of a cell. So if we see different parts of a nucleus, we can see that nucleus has a nuclear membrane or nuclear envelope. Right? It's called a nuclear envelope. The major function of nuclear envelope is to separate the nucleoplasm and the cytoplasm of the cell. So main function is to separate nucleoplasm and cytoplasm. Right, and if you see the nuclear envelope, we can see that it is double layered. So it has a space in between this, right? This space is called perinuclear space. What is this space called? The nuclear envelope or the nuclear membrane is a double layered membrane. It has a space in between and this space is called perinuclear space. Perinuclear space. Right. Now let's go inside the nucleus. We can see thread-like structures. These thread-like structures are called the chromatin or chromatin fibers. Right. These chromatin fibers contains the genes and chromosomes which controls the cell activities, right? So the thread-like structure present inside the nucleus is called chromatin fibers, which contains the chromosome or the genes which controls the cell's activity. That's why it's called the director of the cell, right? Now what else? This condensed structure is called the nucleolus. Now, if we see the nuclear membrane again, we can find that this nuclear membrane is not continuous. This is double layer membrane and it is not continuous. It has gaps in between and this gap is called the nuclear pore. This gap is called the nuclear pore, or we can call it as nuclear pore complex, because many other proteins and carbohydrates, everything is related to here. So we can call it as nuclear pore complex also. So for what this nuclear pore complex or nuclear pore is made up of? Actually, there should be an exchange of materials from the cytoplasm to nucleus and from nucleus to the cytoplasm. This exchange takes place through nuclear pore. Right, because at the time of transcription or translation, first transcription takes place inside the nucleus. The RNA is made inside the nucleus, but if the RNA has to be translated, the ribosome is present in the cytoplasm. So RNA has to be transported to cytoplasm, right? So for that, these RNAs are transported through this nuclear pore. Not only RNA, proteins and many other materials has to be transported, is transported through the nuclear pore to nucleus, and from nucleus also it is transported through nuclear pore. That's all about the nucleus. Now the next option was about what? Let's see. The next option was about the sieve tubes. Sieve tube is a part of phloem, right? So what's the function of phloem in a plant? The main function of phloem in a plant is to transport food or carbohydrate from leaves to different parts of the plant. So the main function of sieve tubes or elements is to transport food materials from leaves to different parts of the plant. Transport food, right? Transport food from leaves to different parts. Now if we see the structure, sieve tubes or sieve elements are straight, perforated, and in between they have a perforated plate between two sieve tubes. Between two sieve tubes we can see a perforated plate which is called the sieve plate. And the interesting fact about this sieve tube is that once they mature, they get denucleated. Once they mature, they get denucleated. That means there is no nucleus present in the sieve tube or sieve element. So we can write no nucleus present. And obviously if nucleus is not present, other cell organelles are also absent in these sieve tubes. But the main difference between sieve tubes and the xylem is that xylem will be a dead cell once it matures. But sieve tubes is still a living cell. It's still a living cell. It's still a living cell. Now, if it does not have cell organelles nor nucleus, how does it get food material to transport into different parts of the plant? For that, it has a cell with it. This cell is called the companion cell. This companion cell gives food materials, energy and other things needed for the sieve tubes through a channel called plasmodesmata. It provides energy, food and everything to the sieve tubes through a channel called plasmodesmata. Through a channel called plasmodesmata. So if we speak about sieve tubes or sieve elements, we can say that sieve elements are living cells, but they get denucleated and they are devoid of all other cell organelles once they mature. But in case of xylem, when xylem matures, they are dead cells or dead tissues. The main function of the sieve tubes or sieve elements is to transport food materials from leaves to different parts of the plant, and they get energy and food for the transportation from the cell near to it's called the companion cell, and the companion cell provides energy and food to sieve tubes or sieve elements through a channel called plasmodesmata. Now the other option was about microbodies. So what is a microbody, or what does microbody means? Microbodies are small organelle-like structures which are found in eukaryotic cells, which are found in the eukaryotic cell. So if we say eukaryotic cell, it's true that it's present in both plant cell and animal cell. So it is present in both plant and animal cells, right? And now what are the different types of microbodies? There are mainly three types of microbodies. They are spherosomes, glyoxysomes and peroxisomes, and these three microbodies arise from endoplasmic reticulum. So the microbodies arise from where? It arises from endoplasmic reticulum. So microbodies are present in eukaryotic cell, both plant cell and animal cell. There are mainly three types of microbody. What are they? Spherosomes, glyoxysomes and peroxisomes, and these three arises from the endoplasmic reticulum. Now if we speak separately about spherosomes, glyoxysomes and peroxisome, these spherosomes and glyoxysomes are present only in plant cells. They are present only in plant cells. But peroxisome is present in both plant cell and animal cell. In both plant and animal cells. They are present in both plant and animal cell. And if we speak the function of each microbody, the main function of spherosome, the another name of spherosome is spherosome. The another function of spherosome is plant lysosome. It's called the plant lysosome. It's called plant lysosome. So you know what's lysosome, right? Lysosome contains hydrolytic enzymes. It helps in digestion. So in plants, instead of lysosome, in the cell where there is more digestion has to happen or more catabolism has to happen, in that type of plant cells, spherosomes will be present with hydrolytic enzyme. It has hydrolytic enzymes present in this. So it helps in the digestion. So it's mainly found in the area where digestion has to happen, mainly in the endosperm of the seeds. Endosperm of seeds. Right. Now we go to the second organic microbody, that is glyoxysome. The function of glyoxysome is beta oxidation. That is beta oxidation. Beta oxidation is nothing but converting the fat into carbohydrate. Fat into carbohydrate. That is, wherever in the cell the food is stored in the form of fat, the glyoxysomes comes into picture and converts fat into carbohydrate. This process is called beta oxidation. So in some seeds the food is stored in the form of fat, such as example is castor seed. Castor seeds, and it is also seen in peanut seeds. Not peanut seeds, actually peanuts. So glyoxysome is present in the cell where the food is stored in the form of fat, and the function is beta oxidation, conversion of fat into carbohydrate. Now the final microbody is the peroxisome, which is present in both plants and an animal cell. But the function of peroxisome in animal cell is not yet identified. In plants, the function of peroxisome is to convert amino acid and uric acid into hydrogen peroxide. So hydrogen peroxide, which is toxic to the cell actually. So it converts amino acids into hydrogen peroxide, H2O2. While H2O2 is toxic to the cell, so H2O2 is again converted into less toxic form or intoxic form. So H2O2 is converted into H2O plus O2. So when amino acid is converted to H2O2, the enzyme is called catalase. And when H2O2 or hydrogen peroxide is converted to water and oxygen, here the enzyme is oxidase, or we can call it as peroxidase. So now we know the function of all the three microbodies. These three microbodies are present in eukaryotic cell. If we say as common, microbodies are present in both plant and animal cell, but spherosomes and glyoxysomes are present only in plant cell. Peroxisome is present in both plant and animal cell, but the function of peroxisome in animal cell is yet to be identified. Okay, now where are the spherosomes present? The another name of spherosome is plant lysosome. So it's present in the cells where there is more digestion or more catabolism takes place, mostly seen in the endosperms, to convert stored food into carbohydrate. Now the function of glyoxysome is beta oxidation, and function of peroxisome is to convert uric acid and amino acids into hydrogen peroxide, and again hydrogen peroxide into a non-toxic form such as oxygen. So here the enzymes that function in peroxisomes are catalase and oxidase. Now let's go back to the question. Now if we see the option, here we have to find out which option is incorrect, right? So first option is nuclear pore act as a passage for protein and RNA molecule in both the direction, that is to the nucleus and from the nucleus. Is this option correct or not? Yes, this is correct, right? We have seen the nucleus and we have nuclear pore to transport materials to the nucleus and from the nucleus, right? So the first option is correct. Now the second option is mature sieve tube elements possess a conspicuous nucleus. They possess a conspicuous nucleus and usual cytoplasmic organelles. Is that true? No, right. When a sieve tube matures, it gets denucleated and all other cell organelles are absent in a sieve tube or sieve elements, and all the energy and other materials whatever a sieve tube needs is provided by the companion cell through plasmodesmata. So this sentence is wrong. Hope we got the answer right. Now the third option is microbodies are present in both in plants and animals. That's true, right? Microbodies are present in eukaryotic cell in both plants and animal cell. So that's true. And the final option is the perinuclear space forms a barrier between the materials present inside the nucleus and the cytoplasm. That is also very true because it's a double layer membrane which separates nucleoplasm and the cytoplasm. So here the answer is option B because the question is which is the incorrect sentence. So hope all the concepts are clear for you and you got the answer. Thank you for watching. Happy learning.